Can you hear me at the back? Yeah? Okay, thanks. So, hi everyone. I'd like to tell you the story of how I got into India today, which is probably more interesting. But I'll stick to my original title and to share about my experiences. And I'd like to thank the organizers for not giving up on me until the last minute. Okay. And also sponsored by November. So just a little bit about myself, since this is the kind of thing. So I'm a geneticist by training, and I got most of my education in the UK, so bachelor's, MSc, and PhD. I also did my postdoc in the SNIoE Institute, and then thinking about whether I should move back to Asia, I did a 1-year postdoc in Japan. Okay. And so just kind of to lay out, so all the kind of suggestions or comments I will make are based on my 10 years of establishing a lab. It can be a survivor bias, but it's really up to the things I encountered in Taiwan. So I set up my lab 10 years ago, and then just moving towards the kind of tenure track system from the system associate and full professor in academia or Taiwan or National Taiwan University. I'm also a global young investigator at EMBO. Okay. Next slide, please. I guess this is a bit like this application. Okay, so quick lab motivation. I wanted, when I came back, I wanted to use Taiwan as an island system to study species diversity around Asia. I particularly work on microorganisms, fungi, and nematodes. And so I wish to understand the genetics and molecular mechanisms behind this diversity. And hopefully one day I can harness the biodiversity to application and even modulate environment to ecosystem health. So this is really just about my journey of microorganism evolution. So skills I had kind of picked up, I mainly kind of sorted by core skills, domain knowledge, and topics. And I guess the more relevant is the choices that I made. So after my undergraduate, this was the time when the human genome first got published, right? You get loads of data, you have loads of predictions, and I didn't want to work on humans because probably 99.9% of all the scientists are moving to humans when the human genome was published. But I wanted to handle genetics data. So I went on and to do a PhD in East. And after the PhD, it was also the time of next generation sequencing was starting, right? So I went to do a postdoc in the SNIoE Institute. Then when setting up a new lab, I wanted to take all the expertises I have gathered, as well as taking up a new technique that I have not learned, but I would like to embrace, which is the metagenomics or the microbial ecology. So quickly going through, right? So you can see kind of the history. You know, PhD was the capillary resequencing. I guess most of you probably don't remember capillary resequencing, right? So it was just 20 isolates of chromosome 3 data of the yeast species. And the cost around almost 1 million UK pounds. And it took me 3 years to, do blast, sequence, align, and annotate, and analyze. And then, but one thing I took was that you can actually infer biology from sequence data. That's how I believe it. And I wasn't very good at experiments, so I quickly shifted towards the more genetic side. And because of the ability, I was able to be asked to form as part of a collaboration, which started to gather all the yeast species, the Saccharomyces cerevisiae, and then we produced the first population biology of the model species. this was the kind of a pre-run of the 1,000 human genomes, right? And then when I had done my PhD, this was a kind of NGS. I wanted to kind of combine the latest technology to the diseases that actually probably need more effort, right? So this is how I moved to parasite, a totally change of field. And the time it took was, at that time, was trying to merge different kind of sequencing technologies. I don't know if you guys still remember 4x4, but this was a very obsolete technology, right? Okay, so but really all of the previous parasite genomes was about drug predictions. And as an evolutionary geneticist, I actually started to put an evolutionary perspective. How do different parasites actually relate to each other? So this was really about a paper of evolution and biology of parasites, first pinned out and revealed. Thank you. Okay, then again, things start quickly, right? It's not about the genome anymore. It's about more of RNA-seq data moving towards phenotyping, right? Then the paradigm shifts to RNA-seq, and then we have all the interactions, post-parasite, etc. But then I realized up to this point, data will continue to be produced on a much faster speed and much higher throughput, and unless you have loads of fundings like China, it's very difficult to just compete solely on the sequencing capacity. And then sequencing projects will just one day be, can be run by every single lab. So ask the right question. For me at that time, seems to be the most important question, and to define my next career. That's kind of the thing I wanted to ask. And the lab, so I started my lab in 2015. And it was also the time that I've gone through the whole PhD postdoc phase. There are kind of the two kinds of nature in research, right? One is to ask questions from and trying to understand nature. And the others is probably try to ask the question to get into nature, right? I wanted to ask the question from and understand nature, and of course, hopefully, to get into nature as well, right? But I kind of split my lab into two parts. One is really trying to go for the ecological side, where I'm primarily interested, and two is the pathogen evolution, which I'm known of, and I can also get funding from the grant agencies in Taiwan. So the first 5 years, right, I guess most of you are in this place. Again, my primary goals was to finish existing projects, so I still have projects with my former mentors and collaborators, no impact factor here, and fund new ideas, project collaborations, embrace new approaches, and establish a team. I think this is probably the most fortunate and important for me to conduct research. So as I said, I work on quantifying genetic diversity at different time scales, whether this is between species or within a species or in a community, and work with different sequencing technologies. I'm just going to show you some pretty pictures of the ecological systems that we've been through. But the primary basic question is, can we just understand the whole picture? I love taking pictures, but in a microbial sense, this is difficult, right? You don't know where the organisms, how do they interact, and I wanted to know that. And this was my team, kind of combined, the three approaches, and with my coffee and speaking, and then start exploring, right? Of course, forests in Asia, islands, even started to work across oceans or work with new organisms. Recently, we are working with this really cool hydrothermal bed. They can survive in temperature up to 80, 90 Celsius. Right? When I was known about this, I thought, already cooked because they are in red, but actually they are still alive and well. Work with new organisms, and collaborators are essential. So, which is, I guess, the purpose of the meeting today. And get the students that are better than you. I think that's very, very important as a junior PR. Okay, so setup done and ready to go on. But how? So now for the rest of the talk, I'll show you my understanding of fungal diversity, which is just like this slide, right? This is really just very, very little bit of how I know about nature. And this is probably because most of the organisms we work are really just a tiny fraction of the organisms that existed, right? Mainly because they are driven by fund agencies, right? Understanding of model organisms, human disease-oriented organisms. But there's such a big domain out there that is waiting for us to explore. So strategy one, we just tapped into unknown domains, right? And so this is one study that we were interested in, the fungal bioluminescence. We sequenced the genome and we revealed how the evolution of fungal bioluminescence actually occurred in fungi. And this was one of the paper. So the strategy can go like this, right? You establish a genome in non-model organism and you try to go through the alternative route. But this is very hard. I have to admit, I don't get enough funding for this one. So again, you work. My kind of the strategy is still trying to work on model organisms in my laboratory. But at the same time, I wanted to know the ecology and evolution of model organisms, preferably unaffected by domestication. So this is where I bring Louis Pasteur, right? And he had a quote which I really love, like, a bottle of wine contains more philosophy than all the books in the world. And this is the scientist's view of yeast, Saccharomyces cerevisiae, right? A lot of the biology was found from this particular organism. But where is the natural habitat of cerevisiae? That's one of the simple questions. And I'm just going to show you the quick video of kind of a trail that we walk. So this is a typical scenery of a broad deep forest, around 1,500 to 2,000 meters in Taiwan. You do have this kind of forest in India as well, which are primarily dominated by Fagacia or Loraceae species, the oaks, the crocus trees. Okay, so we started sampling since 2016. We isolated the substrates, we culture, we isolate them, and you can see signs of fermentation and the growth. We plate them, we identify them, and we found a lot of Saccharomyces cerevisiae actually live in the forest. And we sequenced in the Taiwanese ones, in contrary to the original hypothesis that they are probably a position in the phylogeny. They occurred all over the place, as shown here in the green. This suggests that East Asia or Southeast Asia has an untapped diversity of Saccharomyces cerevisiae. So in contrast to cerevisiae, okay, I need to speak probably a bit faster. So all the wild strands here have allowed us to really just take it back to the lab and repeat the experiment, but with more phenotypes, right? So this is kind of the strategy I have now, right? So differences in the wild strands, how do they actually contribute with the given system, the knowledge that we have now in science? So finally, just probably the current work. We do a lot of crazy things. And one of my students is fascinated with coniferous plants, right? And as some of you probably don't know, this is Charles Darwin. But later, before he's in his last days, he was so fascinated with carnivorous plants. He actually wrote a textbook. And this is all his drawings, actually. And we do have Drosera, the carnivorous plant in Japan. And by employing metagenomics approaches that you usually do in human microbiomes, we actually find fungi has a totally different microbiome setup than the rest of the plant or the environment, in contrast to bacteria. Okay, I'm about to finish. And we found the fungus, they just live on the glands of the carnivorous plants. So, and we were actually able to reveal the intricate symbiotism of carnivorous plants and the fungus, right? Okay, so just the final slides, right? So kind of the key takeaways, as for me, myself, do realize this is probably a bias, but ask the right question that can keep you going, and probably you still need the relevant skill set to ask the questions. There's no point asking a random question when you don't have the ability to tackle it, right? Or you can ask the collaborator. So always identify the right mentor, colleagues, students, friends, and environment that allows you to conduct all this kind of fascinating researches. So on that note, I'd like to thank all my acknowledgements, all the organizers again, and EMBO for making this trip possible. Thank you very much.